Good morning. Today's topic is about general amino acid metabolism. So, introduction about amino acid metabolism. It plays a main role in synthesis of structural and functional proteins. And the essential amino acids are obtained from the diet. So, a 70-age man has an average protein turnover of 400 grams per day. The same amount synthesized and same amount will be broken down. So the amino acid pool will always be in a dynamic steady state, and the rate of synthesis of protein will balance the rate of degradation, so that the nitrogen balance will be maintained. So this is the picture which will describe about the total amino acid in the human body. The dietary proteins contributes about 50 to 75 grams per day, and the breakdown of body proteins is around 300 to 400 grams per day, and it also undergoes oxidation, gluconeogenesis and non-essential in production of non-essential amino acids. About the digestion of proteins, it is mainly denatured, that is broken down on cooking and therefore easily digest, and all the enzymes involved in digestion of proteins comes under the class hydrolysis, and proteolytic enzymes will be secreted in the inactive form, which is known as zymogens, which will be converted to their active form in the intestinal lumen, and this prevents the autodigestion of the secretory enzymes. So it is initially secreted as in inactive form, that is in zymogens, and it is converted into its active form. We will see the proteins, we will see the enzymes one by one in detail. The first one is endopeptidase. They act on the peptide bonds which is inside the protein molecule, and the group includes pepsin, trypsin, chymotrypsin and elastase. Next is exopeptidase. It acts on the peptide bond located at the ends of the polypeptide chain. They include carboxypeptidase and aminopeptidase. Carboxypeptidase will act on carboxy terminal of the carboxy terminal end of the chain, and aminopeptidase will act on amino end terminal end of the amino acids. About the gastric digestion of proteins in the stomach, as you all know, hydrochloric acid will be secreted, and it makes the pH optimum for the action of pepsin and also activates the pepsin. And the acid will denature the proteins. About renin, which is otherwise known as chymosin, which will be active in infants, and it is involved in the curdling of milk. It is absent in adults, and the milk protein casein is converted into para-casein by the action of the enzyme renin. That this denatured protein is easily digested further by pepsin. So renin also plays an important role in digestion of proteins, mainly in the infants. Then about pepsin, it is secreted by the chief cells of the stomach as inactive pepsinogen, that is in the form of zymogen. So the conversion of pepsinogen to pepsin is brought about by removal of 44 amino acids from the N-terminal end by the hydrochloric acid. The optimum pH for activity of pepsin is around 2, and pepsin is an endopeptidase. It's an example for endopeptidase. And further it catalyzes the hydrolysis, that is breaking down of bonds formed by the carboxyl groups of phenylalanine, tyrosine, tryptophan and methionine. By the action of pepsin, proteins are broken down into proteases and peptones. So this is in a nutshell about the various enzymes and their activity and activity on the particular enzymes. For example, pepsin will act on phenylalanine, tyrosine, tryptophan and methionine, whereas trypsin will act on arginine and lysine, and carboxypeptidase A will act on C-terminal aromatic amino acids, and carboxypeptidase C will act on C-terminal basic amino acids. Then we will say about the pancreatic digestion of proteins. So the optimum pH for activity of pancreatic enzymes, that is around a pH of 8, is provided by alkaline bile and pancreatic juice. and secretion of pancreatic juice is stimulated by peptide hormones cholecystokinin and pancreozymin. These are the two important enzymes which plays an important role in digestion of proteins in pancreas. And it also contains the important endopeptidases trypsin, chymotrypsin, elastase and carboxypeptidase. So the enzymes will be initially in the form of zymogens, trypsinogen, chymotrypsinogen and proelastase. And all the three are serine proteases. That is, the active centers of these enzymes will contain serine residues. Then about trypsin, which is initially secreted as trypsinogen, and it will be activated by enterokinase, that is entropeptidase, which will be present on the microvillus surface of the intestine. Once activated, the trypsin will activate the other enzyme molecules. And it is further activated by removal of a hexapeptidase. Hexapeptidase means it contains 6 peptide bonds from the N-terminal end. And trypsin will catalyze the hydrolysis of bonds, which is formed by the removal of groups arginine and lysine. So that is about trypsin. Then applied aspect, acute pancreatitis. Whenever there is premature activation of trypsinogen inside the pancreas, it will result in the autodigestion of the pancreatic cells. The result will be acute pancreatitis. It is a life-threatening condition. If we are untreated, the patient, we may lose the patient. Then about chymotrypsin. And trypsin will act on chymotrypsin in such a manner that the A, B and C peptides will be formed. These three segments are approximated so that the active site is formed. Thus the selective proteolysis will produce the catalytic site. So basically it is about the proteolysis which produces the active site of chymotrypsin, and it is activated by trypsin. Then about carboxypeptidases. Here also again trypsin and chymotrypsin will degrade the protein into smaller peptides, which will be further hydrolyzed into dipeptides and tripeptides, which will contain 2 and 3 peptide bonds, and further action will be carried out by carboxypeptidases, which will be present in the pancreatic juice. Again it is secreted in the zymogen form, which is activated by trypsin, and they are all metalloenzymes which will require zinc as a cofactor. So next we will see about the intestinal digestion of proteins. So the complete digestion of small peptide to the level of amino acid will be brought out by the enzyme in the intestinal juice, that is in the succus intricus part of the intestine. It is the luminal surface of the intestinal epithelial cells which will contain the enzymes which is required for the further breakdown of the proteins. The first one is leucine aminopeptidase. It releases the N-terminal basic amino acids. The next one is proline aminopeptidase. It plays an important role in removal of proline from the end of polypeptides. The third one is dipeptidases and tripeptidases, which will remove the 2 and 3 bonds from the peptides. So this is a pictorial representation depicting the action of exopeptidases and endopeptidases. Then we will see about the absorption of amino acid. It mainly occurs in the small intestine, and it is a energy requiring process. The transport systems are carrier mediated. They require either they can be either carrier mediated or they depend on ATP sodium dependent import system. And there are 5 different carriers for different amino acids. The first one is for neutral amino acids, second one for basic, third is for amino acids and like glycine, and fourth one is for acidic, and fifth one for beta amino acids. I hope you know the classification of amino acids. Then about gamma glutamyl cycle, it is otherwise known as Mr. cycle, and it is mainly operative in the intestine, kidney tubules and the brain, and it plays an important role in absorption of neutral amino acids, and it is affected by gamma glutamyl cycle. The main role is played by the tripeptide glutathione, which contains gamma glutamyl cystinyl glycine, and it will react with the amino acid to form gamma glutamyl amino acid, and it is catalyzed by the enzyme gamma glutamyl transferase. The glutamyl amino acid will be further broken down to give the free amino acid. So the net result will be the transfer of an amino acid across the membrane. That is the main function of gamma glutamyl cycle. And the transport of one molecule of amino acid and the regeneration of glutathione will require 3 molecules of ATP. That will be the energetics of this particular pathway. It's an important short note, you can expect this. This is a pictorial representation explaining the gamma glutamate cycle. I've already told you. So about the clinical application, so deficiency of the enzyme 5-oxoprolinase will lead to oxoprolinuria, that is excretion of a particular amino acid. And allergy to certain food proteins like milk and fish is believed to be from the defective absorption of partially digested proteins, even in Hartnupp's disease due to tryptophan deficiency, and in immunoglycinuria. So these are some of the applied aspects of proteins. Then cystinuria, where you can expect cystine in the urine, and lysinic protein intolerance due to intolerance to the food particles containing lysine. Then partial gastrectomy and various other causes like carcinoma of pancreas, pancreatitis, and protein losing enteropathy. There will be excessive loss of protein through the gastrointestinal tract. About the intracellular protein degradation, it is mainly by the PEST sequence, that is areas rich in proline, glutamate, serine and threonine will give an inherited problem that will break down the protein very quickly. Then about cathepsins, which is also an important digestive particle of protein. They are 18 in number, designated as A to T, and most of them are active at pH around 3 to 5. Then ubiquitin, which also breaks down the protein. It's independent of lysosomes. It is attached to protein with the help of 3 enzymes, E1, E2 and E3. And proteosomes, here ubiquitin tagged proteins are immediately broken down inside the proteosomes of the cells. So this is the overall picture describing the digestion and absorption of proteins. So the pepsin initially will act, which will break down the protein into shorter polypeptides. Then by the action of the enzymes present in the pancreas, it is broken down into amino acids, which is absorbed in the stomach. And here mainly the hydrochloric acid and pepsin will play an important role in absorption of proteins in the stomach. They are further broken down into smaller peptides, and finally it is absorbed in the small intestine. So this is a picture which explains the entire process of digestion and absorption of proteins. Next we will say about the inter-organ transport, how they are transported within the organs. In plasma, all the amino acids are seen within a level of roughly around 1 milligram per deciliter, except glutamic acid and glutamine, which are present in higher concentrations. And breakdown of muscle protein is the source of amino acid, while the liver is the site of disposal of amino acids. So in fasting state, mainly the muscle will release the alanine and glutamine, of which alanine will be taken up by the liver and glutamine by the kidneys, and liver will remove the amino group and converts it to urea, and the carbon skeleton is used for gluconeogenesis. This is the fate of amino acids in conditions of fasting, and the brain predominantly will take up the branch chain amino acids. So in the fed state, it will be entirely different. Amino acids absorbed from the diet will be taken up by the different tissues. Both muscle and brain will take up the branch chain amino acids, and they release glutamine and alanine. And glutamine will be finally delivered to the kidneys, in which it plays an important role in regulation of acid-base balance, while alanine is taken up by the liver. So the general metabolism of amino acids, the anabolic reactions are the ones where the proteins will be synthesized, and there will be synthesis of specialized products such as heme, creatine, purines and pyrimidines. The catabolic reactions where the dietary proteins and the body proteins are broken down to amino acids. And transamination is a process, it's a very important one, where amino group is removed to produce the carbon skeleton. The amino group will be excreted as urea. So the end product of amino acid metabolism is urea, and the carbon skeleton, which is used for synthesis of non-essential amino acids. And it is also used for gluconeogenesis or for complete oxidation. The other minor metabolic functions like conjugation, methylation and amidation also take place. So this is the amino acid sources, where you can expect the sources from diet and body protein catabolism of body proteins. And mostly it is converted into ammonia. Since it is toxic, it will be converted into urea and excreted. So 57% will follow this pathway. The other 7% will be utilized in the synthesis of plasma proteins and body proteins. The other 22% will be seen in the circulation. So formation of ammonia, formation and disposal of ammonia is another important short note. The first step in the catabolism of amino acids is the removal of amino group as ammonia. Since ammonia is highly toxic, especially to the nervous system, and detoxification, that is removal of ammonia, is by conversion to urea, and which will be excreted through urine. Next is transamination, which is also another important short note. Definition is very important. It is the exchange of alpha amino group between one alpha amino acid and the other alpha keto acid. So one amino acid will react with the second keto acid, which will result in the transfer of amino group between the alpha amino acid and keto acid. So the enzymes catalyzing the reaction are generally known as aminotransferases, and all these enzymes will have pyridoxal phosphate as the prosthetic group. And the reaction is readily reversible reaction. So this is an example in which pyruvate is converted to alanine by the enzyme ALT, alanine aminotransferase, by the enzyme pyridoxal phosphate, and it is converted into glutamate and alpha ketoglutarate. So summary is amino acid 1 will react with keto acid 2, and a new amino acid and a keto acid is formed by exchange of amino groups between them. So what is the significance? It plays an important role in removal of ammonia and is the first step of catabolism of ammonia. It also plays an important role in synthesis of non-essential amino acids. So by means of transamination, all the non-essential amino acids can be synthesized in the body from the keto acids. For example, pyruvate can be transaminated to alanine. Similarly, oxoacylated will also produce aspartic acid. Other example, alpha ketoglutarate is transaminated to form glutamic acid. Then about the interconversion of amino acids, if one is high, the other one is low, the amino group will from the number one amino acid may be transferred to a keto acid to give the other amino acid to equalize the quantity of both. It is called the equalization of quantities of non-essential amino acids. Then about the clinical significance is very important. Aspartate aminotransferase and alanine aminotransferase are important liver enzymes. They are part of liver enzyme panel, and AST is increased in myocardial infarction while ALT in liver disorders. Then trans deamination. It is nothing but the amino group of most of the amino acid will be released by a coupled reaction. It is nothing but trans deamination. Transamination followed by oxidative deamination, that is removal of amino group by using oxygen. So the combination of transamination and removal of amino group by oxygen is known as trans deamination, and it takes place in cytoplasm of all the cells, and the amino group is mostly transported to liver as glutamic acid, which will be finally deaminated in mitochondria of the hepatocytes. Hepatocytes are nothing but liver cells. Then about oxidative deamination of glutamate. It happens only in the liver mitochondria, which contains glutamate dehydrogenase, which will deaminate. Deamination means removal of amino group from glutamate to form alpha ketoglutarate and ammonia. First, the amino acid will be transaminated to glutamate, which is then finally deaminated. That is trans deamination. The rate is about 50 to 70 gram per day. So it needs NAD as a coenzyme. NADP can also be used as a coenzyme. Finally, the hydrolysis of glutamine will yield NH3, but in the kidney it will be in the form of NH4 because it plays an important role in maintenance of acid-base balance. Then about minor pathways of deamination, L-amino oxidase will act on all the amino acid, and it uses flavin mononucleotide as a coenzyme. And D-amino oxidase will also undergo the minor pathway, and it uses FAD as a coenzyme. So ammonia may be formed in the body through minor reactions also, like oxidation of monoamines by monoamine oxidase. The other minor pathways include desulfhydrase and histidine. And it is also produced in little quantities by bacterial putrefaction of GI tract. So finally, about the disposal of ammonia, how it is disposed. The first line of defense is, being highly toxic, ammonia should be eliminated or detoxified as and when it is formed. So every minute quantity of ammonia may produce toxicity in the central nervous system. So it is almost always produced by most of the cells, including neurons. It has to be immediately converted into urea and excreted by the kidneys. So glutamine, which is formed, is transported to the liver, where the reaction is catalyzed by the enzyme glutaminase, and it produces ammonia, and it is immediately detoxified into urea. This is the pictorial representation of the disposal of ammonia, where glutamic acid is converted into glutamine by the enzyme glutamine synthetase, and finally by the enzyme glutaminase it is converted into glutamic acid, and it is excreted in form of urea. So next, about transportation of ammonia inside cells of almost all the tissues. The transamination of amino acids will produce glutamic acid, and glutamic dehydrogenase, already I have explained you, it is finally converted into glutamic acid. And glutamine is the transport form of ammonia. It is very important, and in the intestine to the liver, where the alanine is the transport form from the muscle. So finally it is detoxified into urea by the liver cells, and it is excreted through the kidneys. So urea is the end product of protein metabolism. That is one of the important take home messages. Then we will see about urea cycle, which also comes under the topic general amino acid metabolism. So urea cycle is otherwise known as Krebs Hensley urea cycle. As ornithine is the first member of the reaction, it is also known as ornithine cycle. The other names of urea cycle are Krebs cycle and ornithine cycle. So the two nitrogen atoms of the urea are derived from two different sources. One from the ammonia, the other from the alpha amino group of aspartic acid. That will be the sources of urea cycle. So it is the ultimate route for the metabolic disposal of ammonia, and it is exclusively by the liver, and it is transported to the blood, through the blood to the kidneys for the excretion in the urine. And urea is formed from ammonia, carbon dioxide and alpha amino nitrogen of aspartate, which will require ATP. And the enzymes which catalyze the urea cycle are partly in the mitochondria and partly in the cytosol of the liver. So the carbon dioxide will combine with ammonia, and it will form by the enzyme carbamyl phosphatase synthase 1. It is converted into carbamyl phosphate. That is the first step. Then by the enzyme ornithine transcarbamylase, it is converted into carbamyl phosphate and ornithine into citrulline. That will be your second step. The first two enzymes are very important. They play an important role in regulation of urea cycle. These two reactions will take place in mitochondria. The other reactions will take place in cytoplasm. The third one, citrulline, by the enzyme arginosuccinate synthase is converted into arginosuccinate, and arginosuccinase will convert arginosuccinate to arginine and fumarate. Fumarate will enter the TCA cycle. So urea cycle has a link in the TCA cycle also. Then arginine by the enzyme arginase is converted into urea, and it is excreted through the kidneys. Then energetics, 4 ATPs are consumed in the synthesis of each molecule of urea. 2 ATP are needed to make carbamyl phosphate. One will serve as a source of phosphate. The second one is converted into ATP is converted into AMP and pyrophosphate. One is required to make arginosuccinate, and one is required to restore AMP to ATP. So 4 ATPs will be consumed in the case of in each cycle of urea cycle. That will be the energetics of urea cycle. Then about regulation, mainly by the enzyme carbamyl phosphate synthetase 1, which is allosterically regulated by N-acetyl glutamate, and it is synthesized, NAG is synthesized from acetyl CoA and glutamate by NAG synthase, which will activate carbamyl phosphate synthase 1, and it has no other function also. Acetyl glutamate synthetase, which is another important enzyme, is activated by high protein diet, arginine, more consumption of arginine, and starvation. I have already explained this one. The synthesis of NAG increases after intake of protein rich diet by arginine and during starvation, which will decrease the urea formation. Significance of urea cycle: the toxic ammonia is converted into harmless non-toxic urea, and it also disposes of two waste products, ammonia and carbon dioxide. It plays an important role in semi-essential amino acid arginine. It participates in the regulation of blood pH, which depends upon the ratio of the dissolved carbon dioxide, that is carbonic acid to bicarbonate. The ornithine which is formed in the urea cycle can form non-essential amino acid proline, and it is a precursor for formation of polyamines like putrescine, spermidine and spermine. So it has a variety of biological roles. The normal range of blood urea, finally about the normal reference range of blood urea, which is about 20 to 40 milligram per deciliter in case of adults. Whenever you consume more high protein diet, it will show an increase in level of blood urea concentration. So in clinical practice, blood urea level is taken as an indicator of renal function. So it is one of the component of renal function test, and the term uremia is used to indicate increased blood urea levels. There are various causes for uremia. Pre-renal causes include consumption of high protein diet, any cause of increased protein, during increased breakdown of protein in conditions like trauma and surgery, in starvation, diabetes mellitus, and in any case of impaired renal perfusion, in case of cardiac failure. Renal uremia, any cause that leads to reduced glomerular filtration rates and which will lead to retention of urea. Post-renal causes include obstruction to the flow of the urine, that is benign prostatic hypertrophy, that is enlargement of prostate gland, malignant stricture or obstruction in the urinary pathway. So this is about the inborn errors of urea cycle disorders. The first one is hyperammonemia type 1. The enzyme defect is CPS1, and the product which will be accumulated is ammonia. In case of type 2, again it is ammonia which will be accumulated, and it is by the defectiveness of the enzyme ornithine transcarbamylase. The third one is citrullinemia. The fourth one is succinic aciduria. The fifth one is arginemia. So since the urea synthesis converts the toxic ammonia to non-toxic urea, all the defects will result in accumulation of ammonia, which is known as hyperammonemia and ammonia intoxication. The intoxication will be more severe when the metabolic block will be at the level of reaction 1 or 2, since it will accumulate ammonia. So these are the symptoms of urea cycle disorders. Ammonia intoxication will be there, protein induced vomiting, intermittent ataxia, irritability will be there, lethargy and mental retardation. These are some of the symptoms of urea cycle disorders. But finally, about treatment, low protein diet should be administered to the infants. Food intake should be in frequent small meals to avoid sudden increase in the blood ammonia levels. These are the symptoms of ammonia intoxication. There will be an impaired function of the CNS, mainly the brain. There will be tremors, ataxia will be there, convulsions and lethargy. The other GI symptoms include nausea, vomiting, slurring of speech, blurred vision, and severe cases, if untreated, it leads to death and coma. So reasons for toxic effects will be mainly due to the increased production of alpha-ketoglutarate from TCA cycle intermediates to form glutamine and glutamate. So there will be decreased oxidation of glucose, which is the major fuel of the brain. That is one of the reasons for the toxic effects. The other reason include increased formation of gamma glutamine, that is gamma aminobutyric acid from glutamate, which will lead to impaired neural transmission process. The one more possibility will be elevated levels of glutamine formed from NH4, and glutamate will produce osmotic effect that leads to the swelling of the brain. So these are some of the explanations for toxic effects of ammonia. Then finally about 1-carbon metabolism. So 1-carbon metabolism is nothing but it plays an important role in donating the carbon atoms for synthesis of different types of compounds. The different 1-carbon groups are known as 1-carbon pool, which is known as formyl group, formimino group and methinyl group. The fourth one is hydroxymethyl, fifth one is methylene, and sixth one is methyl group. So here the 1-carbon groups will be transported by the way of tetrahydrofolate, which is the active form of folic acid, and tetrahydrofolate is formed from the vitamin folic acid. So folic acid by the enzyme reductase, it is converted into dihydrofolate, where NADPH, which is a reducing equivalent, is converted into NADP, and dihydrofolate is converted into tetrahydrofolate by the enzyme dihydrofolate reductase. So it is mainly carried by tetrahydrofolate. It can be either attached to nitrogen N5 or N10, or to both N5 and N10 of folic acid. And this is in nutshell about the various carbon groups, 1 carbon atoms. Formyl, it has the structure CHO and it can be either carried by N5 or N10 formyl tetrahydrofolate. Then is formimino, which has CH and double bond NH. Methylene, which has CH. Hydroxymethyl will contain CH2OH. Methylene will have CH2 and methyl will have CH3. So it is mainly from the following amino acid. When the serine is converted into glycine by the enzyme serine hydroxymethyl transferase, there will be generation of 1 carbon groups from tetrahydrofolate. From glycine, catabolism of glycine also you can get tetrahydrofolate, that is methylene tetrahydrofolate, where glycine is converted into amino methyl group and finally it is converted into NH3. There will be generation of 1 carbon groups. During metabolism of histidine also it contributes to N5 formimino tetrahydrofolate through FIGLU, which is the active form of histidine. Formimino glutamic acid. FIGLU is nothing but formimino glutamate. And tryptophan will also donate formyl tetrahydrofolic acid. Apart from these amino acids, choline and betaine also, they are also donors of hydroxymethyl groups. And during conversion of choline to glycine, these methyl groups will be recovered. So this pathway is otherwise known as salvage pathway for 1 carbon groups. So in B12 deficiency, during the deficiency of folic acid will also be observed. So there will be diminished transfer of methyl group from methyl tetrahydrofolic acid. So tetrahydrofolic acid will not be regenerated and this is known as folate trap. So in case of vitamin B12 deficiency, you can expect folate trap. And finally, about utilization of 1 carbon groups, how they are utilized. They are used in the synthesis of purine ring, that is C2 of purine, formulation of methynyl transfer RNA, C8 of purine ring synthesis, the formation of glycine and serine, the formation of choline, even in formation of deoxythymidine monophosphate, which is used as a monomer in DNA synthesis. They also play an important role in trans-methylation reactions, including creatine, choline and epinephrine synthesis. And it is excreted as carbon dioxide. So this is in nutshell about the 1 carbon metabolism. These are the sources: glycine and tryptophan, which is converted into formate. The carrier derivatives are N5 formyl tetrahydrofolate, which plays an important role in synthesis of purine. Then histidine, FIGLU, as I already told you, which is the formiminoglutamic acid, and it is carried by N5N10 methynyl tetrahydrofolate, which plays an important role in synthesis of purine, and serine, which plays an important role in synthesis of pyrimidine, and methionine, which plays an important role in synthesis of adenosine methionine. So these are my references. Thank you for your patient listening.